Today we have Eva Szabo giving the first talk. She got her MD degree from Duke University and then did an internal medicine residency at Bellevue, New York University Medical Center. Then she did a medical oncology fellowship at NCI, and she's doing prevention research ever since. So her title is Chief Lung and Upper Aerodigestive Cancer Research Group, Division of Cancer Prevention. Eva.
Thank you. Okay, this works. Fantastic. So thank you for my, 15th year at TRACO or whatever it is. Then it's about right. So today I'm going to be talking to you about non-small cell lung cancer. I'm going to divide it into about half and half, a little bit about biology, not very much, a little bit about therapeutics, and then about half about prevention and a teeny tiny bit about early detection. So this is a complicated slide, but there's something very easy to see. This green thing, this is global incidence and mortality of lung cancer. This green area here shows that globally, across the entire world, lung cancer incidence is a significant part of all cancer incidence, maybe a tenth or maybe a little bit less. But in terms of mortality, it's really an outsized contribution to global mortality, on the order of about a fifth to a quarter of all cancer deaths. It's a major, major problem across the world. Here in the United States, I've been doing this, actually, we were talking about former colleague Ilona Lanoila. So she and I used to trade off these statistics slides, and they have gone up and up, even as smoking prevalence has gone down, because it takes really a long time to undo that damage. So for this year, the estimate is about 234,000 new cases, 154,000 estimated deaths. And this is the leading cause of cancer death in the United States, which is greater than the next three causes of cancer death added together. Now, the good news is that the death rates are decreasing. This is men, this is women, with really the tobacco control initiatives that have been put in place since the 1960s. It takes a long time. The survival has improved over time. This is now therapeutics that we're talking about, and maybe some earlier detection. I put that in quotes. Used to be about 5% in the 1950s. We're now up to about 18% 5-year survival. And the reason why survival is so poor is because most lung cancer is not discovered early, but it's actually discovered when it's already metastatic or at least locally advanced, giving symptoms, and that is often, usually, almost always not a curable situation. Okay, so the risk factors are actually pretty well known. It's tobacco for 85% of all lung cancer. That includes passive smoking. So if you're a child in a household that has heavy smoking going on, you will have that exposure. Spouses, workplace. A prior aerodigestive malignancy, which often is due to tobacco exposure, is also an independent risk factor. Chronic obstructive pulmonary disease, which is a tobacco-related problem. That's COPD, a little bit different from asthma. But even COPD that is not associated with tobacco, and there is such a thing, even that is associated with lung cancer. So it gives you increased risk independent of just the tobacco exposure. Now, most of the other exposures are tend to be due to workplace exposure. So with the exception of asbestos, that's been known for a long time. And although we think about asbestos and mesothelioma, actually the more common cancer to get from asbestos exposure is actually non-small cell lung cancer. Radon, indoor radon exposure is a risk factor. That's why we all need to get our houses tested before we buy them. least here in Montgomery County, many places. And then a whole variety of other workplace exposures, nickel, inorganic arsenic, which actually can be found in well water in some third world countries and such. There's also a genetic predisposition. This is actually fairly hard to study because of the tobacco, to separate that out from the tobacco exposure. But there are actually now well-documented cases of familial lung cancer, and some of which, and for some of these cases, we actually know what the abnormality is. It's the epidermal growth factor receptor, which I will talk about in a little bit, but the T790M mutation in particular actually occurs in families and is associated with adenocarcinoma, even in never smokers. The nicotinic acetylcholine receptor, various subunits have been, polymorphisms are associated with lung cancer incidence as well as COPD incidence. And so these are some of the better characterized lung cancer susceptibility loci. There are lots of studies out there about how people metabolize carcinogens, and there are relative risks of 1.3, 1.2, 1.5 associated with various gene polymorphisms. But this is a very high-level overview. In terms of what lung cancer actually is pathologically, it is not one disease. Molecularly, it's even more diseases. But pathologically, we tend to divide it into what we see under the microscope. So non-small cell lung cancer is primarily adenocarcinoma, which is about 40% of cases. And those tend to be peripheral. Those are the ones that are also associated with never smoking, but very highly associated with smoking as well. Squamous cell carcinomas are about 20% of cases. They tend to be more centrally located, reflecting the cell of origin there. Large cell carcinomas are a fairly uncommon, it's 10 to 15% of cases, again, tend to be more peripheral. And then there's a smattering of other cancers, carcinoids, which can be benign or can be malignant, sarcomatoid, non-small cells, relatively uncommon cancers. The other 20% of lung cancer is small cell, and I think that that's a separate lecture. So lung cancer, because it is often, usually due to exposures, actually the process occurs over a long period of time. You start smoking when you're a kid, a teenager. You develop your lung cancer when you're 50, 60, 70. So it's a long process. And during this process, there is a progressive development of histologic changes in the bronchial epithelium, hyperplasias, metaplasias, through increasing grades of dysplasia all the way to cancer. There's also an accumulating, there's an accumulation of molecular abnormalities, but this gives us the opportunity to really intervene in along this entire continuum. Now, treatment is the ideal intervention. You know who you're treating, right? Because somebody comes to you, they have lung cancer. The problem is that often it's too late to have a cure, except for the very early localized cancers. Early detection would be another option, which we'll talk about a little bit later. There you can start to, you hope to detect early enough that an intervention such as surgery will result in cure. But it's really the whole process of 20, 30, 40 years of tobacco exposure that gives us the opportunity for preventive approaches, and that is what I will really talk about mainly. But before I do, let's talk about, my slides look a little weird. I don't know whether it's me or something about, but anyway, okay. Let's talk about treatment strategies for lung cancer. So treatment is based on stage, like all cancers. If it's early localized, we're talking about surgery. If it's slightly more spread, so it's a bigger cancer or it involves lymph nodes, then we add some chemotherapy to get a little bit of benefit. If it's already spread to lots of lymph nodes or crosses to the other side of the mediastinum, then we're really talking about radiation together with chemotherapy, sometimes with some surgery as well. And that is a much less curable situation, from 5 to 20% 5-year survivals. And once you're out of this central mediastinal area and wherever the cancer originated, if you involve the pleural surface or distant sites, then you're really talking about metastatic disease, which even today is considered an incurable situation. And there we use chemotherapy, immunotherapy, targeted therapies. Radiation is needed for local control. Sometimes we take out some isolated metastases, but it's really a systemic approach. For small cell, it's systemic from the get-go, plus or minus radiation. So, the hallmark, the standards for metastatic treatment of metastatic disease, I have to say, have increased and changed drastically during my 29 years in medical oncology. Chemotherapy is still something that the vast majority of patients will see. We use platinum, cisplatin, carboplatin, with additional drugs. Intravenous treatment goes to the whole body, theoretically. It's used in adjuvant treatment, but mainly for metastatic disease. We now have targeted therapies, which really are for a minority of patients who have specific mutations, and drugs that target those specific mutations actually have very good response rates, but not cure. These are generally oral therapies. They're better tolerated, and they do extend survival. And there's a number of them, which I'll describe. And now, over the last few years, we have immunotherapy, which now has a definitive role, both as frontline and second-line treatment. So personalized therapy, as you see, it's the same graph here, but added to the side. So as we've been with the TCGA and lots of hard work, we now know that in adenocarcinomas, which are 40% of lung cancers, there are a number of different driver mutations which are responsible for this subtype. And a number of these are actually targetable. So I'll talk about EGFR, ALK rearrangements, ROS1 rearrangements as well. But there's a whole list now of molecular abnormalities that we look for when a person is diagnosed with metastatic lung cancer, because in fact, the response rates, if you treat them with targeted therapies across these various mutations, are in the order of 50 to 80%. So about 20% of all non-small cell lung cancer patients are actually appropriate for treatment with targeted therapies. And this is really, it's the epidermal growth factor receptor story that put non-small cell lung cancer into the precision medicine basket, so to speak. And it's a good story because it was found that with specific drugs, gefitinib, eventually erlotinib, all comers of lung cancer, there's a 10% response rate with these new agents. But it's really very high if people have a specific mutation, the EGFR mutation. And this leads to progression-free survival upon treatment on the order of 10 to 14 months with the first-generation therapies, which is very different from the 4 to 6 months that you get with standard chemotherapy, and median survival, which is now on the order of 2 to 2.5 years. So we have multiple drugs. Erlotinib was one of the most successful first-generation agents approved for first, second, third-line treatment. There are others, afatinib, gefitinib. But now we have a new drug, osimertinib, which is now also approved because it not just targets the usual sensitizing EGFR mutations, but also a resistance mutation, the T790M mutation, which occurs in about 50% of people who are treated with erlotinib or other first and second generation treatments. And so osimertinib now is the new frontline winner, so to speak. And this T790M mutation, if you listen real hard, is one of those rare familial lung cancer mutations. So where it came from, who knows, but it's an interesting story. And so this is now a much better path to go into if you have to have non-small cell lung cancer. Your prognosis is better, and there are multiple different treatments available if you have an EGFR mutation. The EML4 ALK fusion gene is another example of a targeted therapy, and this is one of the, I would say, winners in terms of going from identification of the abnormality to a approved therapy. So this was identified in 2007, so 11 years ago. It actually occurs in about 5% of people. Just like EGFR, it is mainly in never smokers. And there is very striking response to tyrosine kinase inhibitors that target this abnormality on the order of 60% or so. And now we have multiple agents which are approved for frontline treatment, some of which are effective if you've had, for instance, crizotinib first, then you can get ceritinib or alectinib, or now brigatinib. Lorlatinib has just been approved. So this is an active area of research and good longer-term control than with chemotherapy, but still not a cure. ROS1 is a third rearrangement. It is another tyrosine kinase. Now we're talking about less than 2% of all comers with non-small cell lung cancer. But again, highly responsive to oral treatment, in this case with crizotinib, with about a 70% response rate and a long duration of response, not curable. So those are sort of the three most common abnormalities that are found. So the whole treatment paradigm, though, has been sort of turned upside down with the advent of immunotherapy, and I'm sure that that's what you're hearing about in just about every disease that there are lectures about. And in particular, the PD-1, PD-L1 pathway is the one that is very important in the treatment of non-small cell lung cancer, and now actually even approved for small cell lung cancer. So PD-1 is a T-cell co-inhibitor receptor. It regulates T-cell activation, and the goal of inhibiting this pathway is to rev up the patient's own immune system. And so you get a better immune response. And there are several antibodies now that are used and that are approved. Pembrolizumab is approved for frontline treatment in people who have high PD-L1, so the ligand expression in tumor cells. It's a little bit hard to see here, but this is overall response with chemotherapy as opposed to pembrolizumab. And what you're seeing at the tail end is that there seems to be a little bit of a flattening of the curve and a separation that is quite striking of about 20% between those who get pembrolizumab frontline versus those who get chemotherapy only. Pembro can also be given with chemotherapy for those who don't have such high PD-L1 expression, and it too shows a very significant survival advantage. You know, as with all treatments, immunotherapy came into use in second line and third line treatment, and so we now have fairly long follow-up of people. And the reason why it is so exciting is that this is one of the other PD-1 antibodies, nivolumab or Opdivo, compared to standard second-line treatment with chemo docetaxel. And again, what you see out here out to 2 years is this flattening of the curve. And so people are asking, is it possible that some of these patients are actually cured? There are people who are out there for 5 years or more after immunotherapy treatment. And how can we move this curve up and keep it flat like this? So now there are multiple antibodies, PD-1, PD-L1. They all have a fairly similar efficacy. So excitement, but we have all seen these folks here 2-3 years out then get recurrent lung cancer. So as exciting as it is, I think the C word, the cure word, needs to be used very judiciously, and a lot of work needs to be done for treatment. And I should mention that these therapies cost over $100,000 a year. They are not completely benign. They're very well tolerated for the most part, except for those people who get endocrine side effects, autoimmune side effects, and those can be quite significant and deadly. So, you know, it's not a tea party, but very effective therapy that a lot of people are getting. So, if we can't cure or can't cure yet, how else can we reduce cancer morbidity and mortality? And so this is where we now start to talk about prevention and early detection. If 85% of lung cancer is caused by smoking, what's the best way to reduce deaths from lung cancer? Well, if you could stop people from ever smoking, stop the young kids from smoking, or getting them to quit very early on, that would be great. But in fact, smoking cessation, as important as it is for overall health and lung cancer health, is still not, first of all, it's not easy, but it doesn't get rid of all the lung cancer. So this 15-year follow-up of a study done 25 years ago, 20 years ago, the lung health study, which was done in people with COPD, looked at rates of death per 1,000 people a year from lung cancer if you continue to smoke, if you intermittently, occasionally quit and then went back to smoking, or if you quit for good. And you see that with a 15-year follow-up, you can have the rate of lung cancer death by completely quitting smoking. What's interesting is that these same curves at 5 years of follow-up showed nothing. So in other words, if you quit 5 years from now, your risk of lung cancer is not, your risk of death from lung cancer is not appreciably different than the people who continue to smoke. So it takes a long time to see that benefit, which is not to say you shouldn't quit and you shouldn't tell people to quit, but it takes a long time. And so what is going on here, and how can we take advantage of this long time to develop lung cancer so that we can actually undo the damage? And this is where cancer chemoprevention, which is what I do for a day job, is all about. It's the use of natural or synthetic agents or strategies. We include vaccines in this now to suppress or reverse the process of carcinogenesis. So not to treat the cancer itself, but to undo the effects of the process and to regress those existing early precursor lesions, to prevent the development of new ones, to suppress the recurrence of prior neoplastic lesions. And this really was codified by the work of Michael Sporn, who was here many years ago, and in many respects is one of the leaders in this field. So with regard to lung cancer, why would we want to go this approach as opposed to treat? Well, as exciting as the current therapeutics are, metastatic lung cancer is rarely curable. Okay, so I need to update this slide. We're talking about 18% 5-year survival instead of 15%, which frankly is still abysmal. We do know from other diseases that cancer is preventable. The P1, breast cancer prevention P1 trial, the START trial, both showed that with tamoxifen given to women who are at risk, and raloxifene and now aromatase inhibitors, if you give them to women who are at risk but do not have breast cancer yet, you can prevent the occurrence of half or so of breast cancers. And we have lots of animal studies with multiple agents where we can model this. And that long preclinical phase with these increasing histologic and molecular abnormalities in some identifiable populations, smokers, hopefully more identifiable than just smokers. This gives us the tools we need to try prevention. So how do we approach this? How do we identify new agents? Ideally, knowledge of mechanisms. And this is where lung cancer prevention is actually not that far along. In the setting of cervical cancer, we know that HPV is the requisite molecular abnormality, and therefore the HPV vaccine was expected to be highly effective. And in fact, we've even seen that with the almost eradication of precancers after vaccination. But with lung cancer, you have many ways to develop lung cancer, even in smokers and, of course, in never smokers. And so we really need a better understanding of the molecular pathogenesis of this process. We often go to preclinical studies to test whether agents and pathways are worthwhile. And often these are carcinogen-treated animal models, or we also use transgenic models that develop lung cancer. Again, this is a good intermediate step, but all of human lung cancer is never modeled in one animal model. And so there are issues in translation, how to get the right animal models and treatments translated to the appropriate human cohorts at risk for disease from that particular pathway deregulation. We also go to observational epidemiology, various studies. This is how aspirin and nonsteroidals were identified for colon cancer, or this is one of the ways for reducing colon cancer incidence and mortality. And then we also look at other clinical trials and say, what's the effect on lung cancer? Again, this is part of how the breast cancer prevention story came to be. So in lung cancer, we're at a disadvantage because there are multiple ways to get to the development of the process. And we have different progenitor cell lineages. So I told you that squamous cell tends to be central, probably arising from basal cells. Adenocarcinomas are peripheral, often arising from type 2 pneumocytes and from Clara cells. Small cell arises from probable neuroendocrine cells, who knows? So you never know who's going to develop what cancer, and why would we think that a cancer arising from a basal cell would respond similarly to a cancer arising from a type 2 pneumocyte? They're fairly different functional progenitors. We also have to remember that it's a long process, and you have to take into account that things change over time. And so those initial changes, which may give a proliferation advantage, may not be as important once you have additional molecular changes that now make it metastatic and modal metastatic, et cetera. So it may be that we have to deliver different strategies to account for the different pathogenesis of lung cancer, and different according to where along that carcinogenesis process a specific person is. That's a challenge. That's a challenge. We tend to select our cohorts for study based on the type of cancer that we think they're going to get. So for the squamous central cancers, we often study bronchial dysplasia, and I'll show you examples of that. For adenocarcinomas, which are peripheral cancers, they don't arise from the bronchial epithelium. So we've started looking at lung nodules, which have different challenges associated. But we don't know how to study prevention of large cell lung cancer or small cell lung cancer, because we don't know how to find those people who are at risk. And of course, non-smokers, which is the majority of the population, there's no way to identify who's going to get lung cancer unless you happen to be in one of those unusual families that has a lot of lung cancer in it, like the EGFR T790M families. An additional issue to keep in mind for lung cancer prevention is the risk-benefit, or for any kind of prevention, because you're talking about people who are healthy now, albeit at risk for development of cancer later. So the efficacy needs to be balanced very well with tolerability, right? So people will take the medicines, and with major adverse side effects that could lead to problems from other diseases. So one of the stories in cancer prevention is the rofecoxib, or Vioxx story. So this is a selective COX-2 inhibitor. It was a blockbuster drug for Pfizer for pain relief, and it was studied for colon cancer prevention in people who had colon polyps. Turned out to be quite effective, 30% decrease in new colon polyps, but a 2 to 3-fold increase in heart attacks. Those come early. The benefit from colon cancer prevention comes much later. And so actually rofecoxib came off the market because of that study. Now, should have come off the market anyway because of this side effect that was not recognized in the initial shorter duration studies. But the point is that this is a very important issue for cancer prevention. A fourth issue to consider is, for phase 3 studies, we look at development of cancer. But for those intermediate efficacy studies, how do we know that we're being effective? Now, in cancer treatment, we do CAT scans, and we look at the lesions that are the cancer. We measure them, and we say, are they getting bigger? Are they getting smaller? Is there a response? And there is a reasonable correlation between response rate and actual survival. That's actually not as strong anymore in the age of immunotherapy. So there's lots of angst in the treatment field as well. But nevertheless, there is something concrete that you can measure. In cancer prevention, you're looking to measure something that's not there yet. You don't have a cancer you can measure. How do you find the premalignant lesion, which is often very small? You don't do it by imaging, usually, certainly not for lung cancer prevention. So we are still in the process of trying to figure out what are some of these intermediate markers that can give us a sense of preliminary efficacy that could then give us enough information to move on to phase 3. So it's a challenging field, and I'm going to show you how we've been approaching it. And maybe the disclaimer up front that we don't have any approved drugs for lung cancer prevention, although we do for other risk reduction in other cancers. Aspirin is now recommended for colon cancer in people who are high enough risk in terms of cardiovascular disease to warrant treatment for that as well. Tamoxifen, raloxifene are approved for risk reduction for breast cancer. So inflammation is perhaps the single most compelling story, if I may call it such, for lung cancer prevention. It is well known that the inflammation that you get from tobacco exposure is important in the genesis, actually, of many cancers, lung cancer in particular. And we have a large body of data showing that intervening in animal models that are primed to develop lung cancer with oral or inhaled steroids can, to a great extent, reduce the number of tumors, the size of the tumors, extend animal life. There is some epidemiology from some studies in cohorts that have COPD and were taking inhaled steroids to show that those people who take inhaled steroids compared to those who do not have a decreased incidence of lung cancer. This is an example of an animal study, and what you see is this is a steroid given orally called budesonide. Increasing doses give you a 80% decrease in the number of tumors these animals that are exposed to a carcinogen will get. And not only do you have fewer tumors, but you have less advanced tumors. So this black is a long duration of treatment, and the number of percentage of tumors that are carcinomas goes down, whereas the percentage of tumors that are adenomas, so not yet as advanced, goes way up. So you decrease the total number and you shift to a less advanced histology. So how do we translate this to human beings? So back in the late 90s and early 2000s, when this field really came to be in its own, the only way we could translate animal studies would be to look at bronchial dysplasia, which is a precursor and a risk marker for lung cancer. It's part of that genesis from starting with a normal epithelium to increasing grades of dysplasia. As you can see, it's looking worse and worse until you get to carcinoma in situ. And to make a long story short, just focus here. I give you lots of numbers here, but people with low or high-grade lesions who were followed up over an extended period of time, about a third of them developed invasive cancer, about 40% of which were from the abnormal site that was being followed versus the other 60% being elsewhere, either in the bronchial epithelium or in the periphery. So the presence of bronchial dysplasia identifies a very high-risk cohort, and some of those lesions will progress onto cancer even in a relatively short period of time. So the way the studies were done, therefore, and this is a trial of inhaled budesonide based on those animal models, was to look at people with bronchial dysplasia, do a bronchoscopy, put a tube down, do a biopsy, count the number of lesions, take them out, look at them under the microscope, give the budesonide or a placebo for 6 months, and then to look at the number of residual bronchial dysplasia sites, the grade of the dysplasia, as well as a number of other markers. These are hard studies to do. Stephen Lamb at British Columbia is an incredible investigator who was able to do a number of these. And so in our inhaled budesonide study, which was again a direct translation, we screened about 1,000 people with sputum cytology, identified those who had abnormalities. About half of those individuals underwent bronchoscopy. Ultimately, 112 smokers with dysplasia were randomized to budesonide or placebo. Now, unfortunately, the study was negative, hard to do. 6-month intervention may or may not be enough, but the complete response rate versus progressive disease response rate were not any different. So the animal studies I described looked at adenomas in the peripheral lung. This was looking at squamous cell precursors, bronchial dysplasia. This is about the era when computed tomography, CT, started being used for started being studied for early detection of lung cancer. And as part of this study, people also underwent CT, and there was this slightly statistically significant, but some decrease in the number of lung nodules that resolved. That would be the part of the lung that would be more relevant to what the animal studies actually showed. And so our second study, and this is again, the trial model is under evolution. The second study that we did with inhaled budesonide was to look at people who have CT-detected lung nodules, actually ones that were persistent, because these nodules can come and go. They were being studied for early detection of lung cancer with our colleagues at the European Institute of Oncology in Italy. And so we randomized 202 participants who had these nodules to inhale budesonide and placebo now for a year, because that's the interval between their CT screenings. And this study is giving us a hint as to how maybe we should do the trial. So I told you there were 200 participants. 70% of them had small solid nodules. Whether you gave them budesonide or placebo, no change. So these were probably little areas of fibrosis or whatever, and they really were small. They were under a centimeter. On the other hand, the ones that were not solid, they were what we call ground glass opacities, there seemed to be a difference with a bigger change in those who got budesonide, and the same with the partially solid nodules compared budesonide versus placebo. But this was clearly not statistically significant. This was 30%, 60 people altogether out of the whole cohort. When we followed these people over time, because they were part of the CT screening project, they got 1 year of treatment, and then they were off treatment. Interestingly, the budesonide treated participants, the nodules continued to get a little bit smaller, to the point that at 5 years, this was statistically significant. What to do with that? Our take home was, don't study these solid nodules, study these other nodules. Now, what are these other nodules? Well, they look like this. These are the ground glass opacities, which over time we're starting to get a better understanding of the natural history, although only from the ones that are large enough to be taken out. These are little indeterminate nodules. Some of the studies where they have been followed over time and taken out show that about a quarter to a half of them are this lesion here called atypical adenomatous hyperplasia, which is a precursor to adenocarcinoma. Some of them are bronchoalveolar carcinomas, minimally invasive carcinomas, but there is a reason to think that some of them are precursors to adenocarcinomas. So here is one recent study of the natural history of these nodules. 795 patients, so it's a pretty large study. 1,229 subsolid nodules, so these ground glass opacities, either with completely ground glassy or with a very small component that is solid. And these were followed over time, okay? And the bottom line is that over this 4-year or so period, average period of follow-up, about 1% of them actually became invasive cancer. Another 3, 4, 5, 6% were either minimally invasive or adenocarcinoma in situ, and a small number of them were actually, upon being taken out, were atypical adenomatous hyperplasia. So some of these really do progress. They progress over time. Here's an example from my own practice, from our own clinic. This is actually a family friend who came to us in 2011. She went to the emergency room because she had shortness of breath. They did a CAT scan, try to make sure that she didn't have a pulmonary embolus, and they found this little nodule right here. Actually, there were 3 or 4 of these elsewhere as well. So they told her, come back in 3 months. Here it is again, not much different. It looks a little bit less prominent, but it's really about the same. Then they said, come back in a year, and like all good people who feel good, she said, yeah, I will, and then forgot about it until 2010, when she again had shortness of breath, went to the emergency room, had a CAT scan, and this time it looks like this. Still, still ground glassy in appearance, but now bigger. So this time around, she did come back a year later, and now it's got a solid core, and it turns out to be a stage 1 invasive adenocarcinoma with areas of adjacent atypical adenomatous hyperplasia. So it takes a long time. I put it here. I show this here because this can happen over a long time, and these ground glass opacities do need to be followed long term. We look back at the National Lung Screening Trial as to what happens to people who have only these ground glass opacities, and we found that early on, very low risk of cancer compared to those who have solid nodules, but 5 years out and more, there's about a 3-fold increased risk of cancer. So some of these nodules are clearly precursors, and it takes a while for them to develop into invasive cancer. Studies are now being done looking at the molecular profile of these lesions, these adenocarcinoma precursors, and these early non-invasive or minimally invasive adenocarcinomas. And like everything else, they are complex. They are not simple genetically. So even early on, these precursor lesions have multiple abnormalities. One thing that was interesting in this huge effort, but it wound up being 6 patients with AAH only, although 5 of them had a smoking history. These AAH precursors had lesions that are often not shared, abnormalities that are not shared with the tumor. Now, it's not the precursor to that tumor that is in the patient from whom it was resected, but maybe in some cases they are related. And what was particularly interesting is that a lot of these AAHs in this particular study and in other studies as well had BRAF mutations. BRAF is one of the known drivers. It is one of our targeted therapies, but it's quite uncommon. And so this suggests that BRAF alone is far from sufficient. In fact, it's not what leads to lung cancer in most cases. So there's a lot of heterogeneity in these premalignant lesions. And the reason I stress this is that we need to understand what's going on, and that is how we will make progress in identifying targets for lung cancer prevention. I'm going to tell you a couple of other stories, moving along quickly. A number of years ago, a meta-analysis was done. Actually, it was a follow-up of a number of studies using aspirin, mainly for cardiovascular reasons, and looking at the long-term follow-up. This is by Sir Peter Rothwell et al., whole series of papers, about a 20% decrease risk of cancer death, which you see really starts to happen after 5 years or more of aspirin. And of particular interest is lung adenocarcinomas, where these 2 curves really start to diverge fairly quickly within about 2 years, and a quite significant decrease in lung adenocarcinoma, but not squamous cell carcinoma death. And so this became the source of our next CT screening nodule trial, which we're waiting for the final analysis now. So I told you that we looked at all CT nodules, gave people inhaled budesonide. Now we focus only on the non-solid and part-solid nodules, the one that seem to modulate with inhaled budesonide. And so we took asymptomatic current and former smokers who were undergoing CT screening, had these persistent non-solid and part-solid nodules, and gave them a low dose of aspirin versus placebo for a year before their next CT scan, and next year I'll have the results of this trial. But this is our current model for studying lung adenocarcinoma prevention. I'm going to tell you one last story very quick because it shows how we try to get into using the current omic technologies to really learn a lot more. So myo-Inositol is a substance, a drug, which is really an isomer of glucose. It is a very active signaling molecule and actually comes from diet. It's been studied in a number of different conditions. And there have been a number of studies looking at myo-Inositol and carcinogen-induced animal models, all of which showed a significant inhibition of carcinogenesis, including a model of smoke exposure, which is actually very difficult to do and generally negative for most other agents. So this drug, as I said, has been used in multiple short-term studies, but significantly, it is what we call GRAS, generally regarded as safe by FDA terminology, which means that you can just give it, you don't even have to have an investigational new drug application, and it makes you feel good that it'll be tolerable without the rofecoxib long-term side effects. So again, with Stephen Lamb, we performed a phase 1 study looking at the maximal tolerated dose in 26 participants who had bronchial dysplasia, found a dose. This was an uncontrolled study, but there was a 90% decrease regression of dysplasia compared to about 50% in historical controls. Not the way to do a study, but for phase 1, it gave us a sense that perhaps this could be effective. And significantly, we teamed up with Avi Spira in Boston University, who had been studying the abnormal pathways that get activated in the airways in the field of smokers. And he found that the PI3 kinase pathway is activated not just in people who have a concurrent cancer, one that doesn't necessarily have PI3 kinase mutation, but also in people who have a dysplasia someplace along the bronchial epithelium. And furthermore, in that small number of people who we treated with myo-Inositol, 9 out of 10 of whom had regression of dysplasia, they also had reversion of this PI3 kinase activation signature. So what does this mean? Is this a way to identify the smokers at increased risk for lung cancer? Because we know that those with dysplasia are at increased risk. Certainly easier to get normal brushings than to identify dysplasia from biopsies, which are very discrete lesions, and you don't remove the biomarker with your brushing. So this is potentially a good cohort to study and potentially a new clinical trials model. So what do? We went to a phase 2 study using myo-Inositol, same way as what I described with bronchial, with the inhaled budesonide. The bottom line is that about 40 people were treated with either myo-Inositol or placebo, all had dysplasia at baseline. We were looking at dysplasia at follow-up. And unfortunately, this study showed a very heterogeneous outcome. There was a non-statistically significant, so there's no statistical significance in any of this, but there seemed to be a slightly increased incidence of regression, complete regression with myo-Inositol, but also increased incidence of progression. I think that that's probably too fine a light on all of this. The bottom line is that there was no statistical significance. What about our biomarker, the PI3 kinase AKT pathway? Still a correlation with response, but I don't know what to do with that because that doesn't tell me anything. If responders have this, but it doesn't tell me who's going to respond, it's only an understanding of the effects of the drug. It doesn't help us move this drug forward. So altogether, this was another negative study, but it showed us the power of using omic technologies, and that's where we need to be moving forward with. Okay, I'm going to spend just 5 minutes, which is what we have left, on a huge field, early detection, but you need to know only a few things right now. Issues in early detection. There are biases. Sounds easy, right? We find it, everything, you get it out early, all is good. Not that easy. There's something called lead time bias. You are screening, you find cancers earlier, but in fact, if you hadn't found them, the person would die at the same time. Okay, so survival will appear longer, but in fact, person dies when they were supposed to die no matter what. This is why you don't look at lung cancer specific mortality, you look at overall mortality. Length bias. You're more likely to find the indolent disease with a longer preclinical phase than those really aggressive tumors that develop between screenings. So these are the better prognosis tumors, they have a better outcome, but again, in the big picture, you're not helping the population overall. And finally, overdiagnosis, where you identify clinically unimportant lesions. They look like lung cancer. They would have never led to death from lung cancer. This is particularly an issue in prostate cancer screening, but actually occurs in lung cancer too. And of course, once you put people into the medical system, you're going to have morbidity, you're going to have mortality from the screenings, the follow-ups of the screenings, the cost of the screenings, et cetera. So, few things to know. Chest X-ray has been studied many times, unclear answers. This is the definitive study, the PLCO, 154,000 participants. Chest X-ray versus usual care, no difference in survival. So chest X-ray alone has been not used for a long time, used to be used in the 70s, 80s. It is not the thing to do for early detection. The National Lung Screening Trial, again, huge, 53,000 smokers, 30-pack years, underwent CT screening, this helical CT screening versus chest X-ray, 3 times altogether. And this was a very significant positive. Lots of people had positive test, many more than with chest X-ray, but a 20% reduction in lung cancer mortality, not just incidence, but mortality, and actually a significant reduction in all-cause mortality, reflecting that lung cancer is a major cause of all mortality. This is just a couple of the graphs showing this is cumulative number of lung cancer cases. So this is X-ray, this is CT, and of course you identify more cases. But what's really important is the deaths from lung cancer, low-dose CT versus chest X-ray, 20% fewer. And although there have been a number of studies in Europe, the most recent data from the Nelson trial in the Netherlands is the first large study other than NLST that was once again very highly positive. That's not been published yet, so I don't have the graphs, but it was presented at a meeting in September. So lung cancer screening is here to stay. How do we make it better? Obviously there are lots of nodules. Most of those people don't have lung cancer. Can we use that? How do we better use it to feed into our chemoprevention trials? So this is one study from McWilliams and colleagues and Stephen Lamb, our collaborators, showing that using a variety of factors, age, sex, family history, emphysema, COPD, as well as nodule characteristics, you can identify a higher risk cohort that is most likely to develop cancer. And so these are the people who we need to target for preventive approaches. All right, so how do we move forward? We need to understand biology. I showed you a lot of negative studies. Part of that is because we don't understand the biology and the natural history. We are working on moving forward the premalignant, the a Precancer Atlas, which is like the large TCGA effort, but now focusing on those earlier stages of carcinogenesis. We need to use agents that are appropriate for the pathways that are likely to be deregulated, drugs that are safe, for instance, repurposed drugs. And we're now starting to move into harnessing the immune response as well. It's important to know which of these premalignant lesions will progress. Who will progress? Why? And that'll tell us how to intervene. So, innovative trial designs using omic technologies like what we discussed, focused on molecularly homogeneous cohorts, which we haven't done before. It's a challenge. And getting multiple trials to actually build a body of evidence that will get us to phase 3. Summary, last slide. We have made a lot of progress in understanding lung carcinogenesis. Precision medicine is applicable to a significant but small subset of advanced stage patients. We need other approaches. These are the early days of immunotherapy. Prolonged survival, again, in a small subset. And we need to learn how to apply this to prevention. That's not yet clear how to do that. Early detection with helical CT is here to stay, decrease lung cancer and overall mortality. And these new targets and tools are now available for studying early disease. And so over the next 5 to 10 years, maybe we'll have some studies that will be positive to present to this forum. All right. I'm done. I don't know whether there are any questions.
Jun Wei, who's giving the next lecture. And he got his Ph.D. in Baylor College of Medicine. He did postdoctoral training in Paul Melch's lab in the cancer genetics branch. And then he joined the oncogenomics section and became a staff scientist in 2009. He assists Javed Khan. And he's going to talk to us today about applying genomics to precision medicine. June.
Okay. Can you hear me? Good? Okay. Thank you, Terry, for inviting me to give this lecture. And here's the outline for what I'm going to do for the hour. I'm going to give you some review of the success and the challenges of treating pediatric cancer. The reason I'm doing this pediatric cancer is our section has a long-term interest in the pediatric cancer. But the trend of the pediatric cancer actually is the same as the adult cancer, the challenges and some success. And the next section, I'm going to talk to you about what is genomics. And especially there's a technology called the next generation sequencing, which is the main working horse for the genomics now. I'm going to introduce you that technology and then give you a study that illustrates how to use these genomic tools and genomics to do precision medicine. Okay, this is a slide shows that over the past several decades, and actually there is a very successful story to treating pediatric cancer. You can see this graph, the survival rate is drastically improved in all sorts of cancers in pediatric from the 60s to 90s. However, the challenge is, seems this kind of improvement in treatment is reached to its plateau. And this is a mortality rate over the several decades. You can see it's keeping decreasing until around 2000, and it's level off. So we are reaching to a treatment plateau. So all the existing treatment seems to not reduce further the mortality rate. So that's the challenge of the treating the pediatric cancers. And really the challenge actually is to rely on those specific type of disease. Specifically, it's metastatic and recurrent disease, and or it's refractory cancers that is resistant to current therapy. You can see all different kind of cancer. It's all this high risk metastatic disease, stage 4 disease. They have very poor survival for all the cancers. And this is remain true in the adult cancer. If you have a local disease, the chance of being cured is relatively high. But if you have a metastatic disease or disease come back, relapse setting, and this usually is true for all the cancers. So let's think about the biology of cancer, okay? And this slide just give you illustration how important the gene expression is in the biology. So this is a very dramatic consequence of gene expression. You can see this caterpillar and this butterfly, they are all share the same, exactly same genome. However, it's because of the gene expression and the expression pattern and resulting in the different proteome and the different resulting in different tissue, and then have different physiology, and then get a totally different phenotype. And this is true for human being too, right? And we know that all our cells share the same genome, same DNA. However, because of this expression, different pattern, the timing, the location, result in all sorts of different kind of tissues to form our body, and they all have specific function. So this is the central dogma of the biology. I'm pretty sure you're familiar in like a middle school or college textbook. So the information flow in any biological system is from the DNA towards the protein, right? So this is the genomic DNA, and through the transcription, we make the messenger RNA, and then the messenger RNA translate into the protein, and the protein is the one to give the phenotype, and this is including the cancer phenotype, diagnostic of cancer, and the response to the treatment. This is all result of the protein. However, the system is not that simple. Actually, it's very complicated. You can see there's a lot of feedback loops to the protein will regulate the mRNA, messenger RNA. Also, the protein will regulate the DNA. And there's another group of RNA called the microRNA. Was discovered fairly recently, about like 20 years ago. And we found that these microRNA, these about only a couple of thousand, in the number of thousands, but they have very crucial regulatory functions for the translation process and the mRNA stability and the transcription process from the DNA to the messenger RNA. So this is a very complex system, and this is probably only the surface that we know. There's a lot of things we don't know yet. And in terms of numbers, there's a 3 billion base pair of DNAs in our genome, and these will transcribe into about 20 to 30,000 genes. But on the top of that, because the genes were eukaryotic system, so it got spliced. There's an alternative splice form. So although we only have 25 to 30,000 genes, but the resulting, the alternative splicing, the transcripts can be more than like 150,000. And as I told you, there's about like 1,000. I think this number is probably not accurate. It's a couple of thousand microRNA. And also there's non-coding RNA. It's a larger than 20 base pair. There's microRNA is about 20 to 22 nucleotide length of RNA. but there's a longer coding, non-coding RNA. There's a lot. It's still in the stage of being discovered. And on the top of that, there is about 500,000 proteins made during this this process. So you can see this is the numbers that we're talking about. And also in the field, usually this is the boundary, the study of this category, the DNA and messenger RNA and the microRNA is called genomics. And this domain to study the protein and its modification is called proteomics. So majorly we're using the genomic tools. So my talk basically is concentrated on this kind of this side of domain. And the recent the study shows about 80% of the genome actually they are transcribed, so they are functional. Before we think the coding gene is the major important component of the genome, but it only consists about 1 to 2% of the whole genome. However, recently data shows about 80% of genome is functional, they're transcribed. So the majority of genome actually is devoted into the regulation of when and where genes are made and the protein expressed. So the challenge of genomics is how to measure or detect genes and their products in a massively parallel way, right? Because we have such a big number of genes that we needed to measure or detect. So the requirement for this kind of process is using the high throughput technology. We cannot use Northern to study all the whole genome because that's just too low throughput. And if we have a high throughput technology, we have to have computational power. So these two are the combination that result in a very landmark scientific project, which is a human genome at the turn of the century. Actually, it's the end of last century. It's like from 1990 to 2003, the NIH lead sequencing of the first human genome. You know, spent about 13 years, a couple of $1 billion to sequence one genome. So, but this is a very important scientific discovery because it's map out the genes and on the chromosomes, so give us a map that we can do a lot of cool things easily and fast with nowadays technology. I will talk about in this talk. So this is the first generation genomic tools. It's called a microarray. I don't know how many people hear about it. And the microarray basically is using all different technology to print this probe, a nucleic acid probe, onto a solid surface so that you can use micro hybridization to detect your gene expression. Okay, that's the concept behind it. And there's a different way to make a microarray in the old time. You can use mechanical quill pen, just dip into the DNA solutions and you print on the surface of the glass. Or you can use inkjet technology. This is electronic piezo. The printing head can, just like the inkjet printer, okay? Instead of printing ink, you use the probe to print on the slides. And then later they have technology use a lithographic mask, which basically use the semiconductor production methods to grow the nucleotide on the surface of the silicon wafer. And the latest method actually they use, it's using the technology called the digital micro mirror device. It's the same device like in this projector. It can, it doesn't need the physical, the light mask to make those chips. Instead of they just use this micro mirror device to direct the light to synthesize the nucleotide on the surface of glass. So when the microarray was made, and we can extract the biological samples, for example, the RNAs from healthy individual and cancerous tissue, and we can label them with a fluorescent label and put it into the hybridization chamber. This is the microarray chip, and you put these things in, and the hybridization happens here, and you use a scanning microscope to scan the slides so that you can detect the signals on each spot. For example, this spot means there's more cancerous signals bind to this spot, this probe. But this green spot means there's a more healthy messenger bind to that spot, okay? And you can quantify it and to measure the signal of the in those original samples. So here I give you one example to use such technology to do this kind of genomics. Nowadays, this is kind of a crude way, but at that time, it's a state-of-art way to do things. So in this case, there is a kid that was referred to the POB, the Pediatric Oncology Group, the branch, to have a second opinion of this tumor. So it's a pretty big mass happening in the upper pole of the left kidney here, and also have a mass in the inferior vena cava. So at that time, the diagnosis of the initial diagnosis, they think is a Wilm tumor, which is a kidney tumor, okay, in these kids. However, when they treat as a Wilm tumor, but the response was not that good. So that's the reason they come to the POB to ask for the second opinion. So we took the biopsy sample, we extract the RNA from it, and we put it into the microarray. And we did a gene expression profile of this tumor, and we did a principal component analysis with the other tumors. And you can see this tumor here is clustered with neuroblastoma instead of this yellow, which is Wilm's tumor. So from the gene expression profile, we can diagnose this is not a Wilm's tumor. Instead, it's a neuroblastoma. So lately, this kid was transferred into the treatment, put on the treatment for neuroblastoma, and the response well for this treatment. So this is the first time we demonstrated this is a very powerful technology we can use directly for the clinic. Now, there's nowadays, the genomic tool majorly we're using in the lab is using this technique called the next generation sequencing. And so this is the way how it works. Basically, it's a shotgun sequencing, okay? We take the genomic DNA or RNA, and we do a fragmentation, and we do a site selection, put an adapter on it. We just put on sequencer to sequence every bit of the DNA on the sequencer. And then we can use the reference map genome, which is the map generated from the Human Genome Project. As a map, we can map all those fragments to the genome, and so that we can know where those DNA or RNA come from, right? And we can put back the original sequence of this sample. So that's basically the principle behind this technology. So this is a picture show you, this is a real picture from the sequencer. And this is a field, a picture from the sequencer. You can see each dot represent one Sanger sequencing. So each dot is a little bit unique DNA fragments. And you can see these millions of dots on this field. And this is the scale. Okay, what you saw that is just a little square on this big chip. And this is the flow cells and the chemistry happening in those flow cells. So this is a massive parallel sequencing. One experiment, you can sequence like millions, hundreds of millions of DNA fragments. So you can rapidly sequence the human genome. Nowadays, we can sequence the human genome overnight, okay, instead of spend 13 years like the first human genome. So what kind of information we can get from this kind of experiment? Okay, if we do the genomic DNA, we can detect the point mutation, we can detect the indel, and we can detect the homozygous deletion, because if you have a deletion of the material, you just don't see it, okay? And you can see it's a homozygous deletion. You can see some, but the coverage is lower than it should be. And the copy number gain, and interestingly, we can, if we do the whole genome DNA sequencing, we can detect the translocation breakpoints. This is all in the base pair resolution we can detect. And in some cases, if the tumor is caused by the virus or whatever, we can detect the pathogen DNAs in this kind of experiment. And we can also sequence in the RNA. This is called the whole transcriptome RNA sequencing. And the information we can get is we can get the digital gene expression. We can get the expressed mutation, detect the mutation in the sample. And we can detect the fusion transcript. That's a very important aspect of this, this type of experiment. I will show you some example later. And we also can detect the alternative splicing event and the RNA editing, novel transcript, and also non-coding RNA. So what is the advantage of this sequencing technology? First is it does not need to prepare clones for DNA fragments, which microRNA needs it. And then another thing is there's no need of prior knowledge for probe design, because if you do the microarray technology, you need the probe, then you need to have a prior knowledge to design the probe. For example, if you have a fusion gene you want to detect, if you don't know the fusion, you cannot just design the probe easily. But in the sequencing experiment, you don't need that prior knowledge. And we can also detect the balanced genomic structure change that is unable to do in the microarray. And this parallel sequencing is at the base pair resolution immediately can tell you all those detailed change. And it's a very high throughput. Another advantage of next generation sequencing is this platform can acquire all different kinds of genomic and epigenomic information. So showing these slides, okay, we can sequence DNA, RNA, and we can have whole genome sequencing or the whole exome sequencing, which only do the genome partition, pull out the coding region to sequence it. Or we can pull out the protein binding places, the DNAs. This is called the, we can use to detect the methylation or specific like transcription factor binding site. That's called ChIP-seq. So all this kind of thing, we can having the information of the copy number change, gene arrangement, rearrangement, and the methylome and the epigenome and the damaged mutation. We can have all those information with the same kind of platform. And the same with the RNA. On the RNA side, we can detect the messenger RNA, non-coding RNA, and all those have gene expression. All this information we can have. And use the same platform, we can detect the biomarkers for diagnosis and prognosis. And also we can study biology with that to find out what is the driver event for the cancers. And hopefully we can use this kind of information to develop therapeutic targets. So this slide shows the the challenge of pediatric cancer. Because after the next generation sequencing technology is widely available, and a lot of, in the past about 10 to 15 years, there's so many studies to study all different cancers. But we see there is very peculiar things about the pediatric cancer. You see, this is all the adult cancer. This is measuring the somatic mutation burden. That means how many somatic mutation per megabase, how many somatic mutation are there in each cancer. You can see adult cancer generally have more mutations than pediatric cancer. Pediatric cancer, very few mutations per cancer. So that will give us challenges how this genomic can help the clinical care for this pediatric cancer. Because we don't have too much directed target that we can do for the pediatric cancer. So in order to find out if this is useful technology, that we did an experiment, a study to use genomics to study pediatric cancers for the patient coming to the CCR, for all the patients coming to the CCR. And in this case, we use a state-of-the-art multi-dimensional integrated omic study platform. So this is a study design. And this is a pilot to determine the utility and the feasibility of performing comprehensive genomic analysis to identify the clinically actionable mutation in pediatric and young adult patients with metastatic, refractory, or relapsed solid tumor coming to NIH. So in this study, we recruit 59 patients, and they are age range from 7 months all the way to 25 years. And there's like 20 different diagnosis category, including most of them is solid tumors, that is not central nervous system tumor. And we perform the multi-omics. Basically, we use exome. Basically, it's the sequence of all the coding region of the genome in germline DNA and the tumor DNA. And also, we combine with RNA sequencing of the tumor, and use the Illumina Omni SNP array to study the copy number of the tumor. So first, we have to define what is considered as actionable. Okay, there's two sides of thing. One is the actionable germline mutation. It means this is a hereditary part of the patient. So our definition is the germline mutation, actionable germline mutation is a loss of function mutation on non-hotspot activating mutation of a cancer consensus gene, which this is a collection of the Sanger database, or the pathogenic or likely pathogenic mutations of American College of Medical Genetics genes. So there's a list maintained by the American College of Medical Genetics. They have a gene list saying that if you find any mutation from these genes, you are obligated to report, okay? Because those are very serious mutations like BRCA1, BRCA2, TP53. Those have a very dire consequences, not only for the patient, also for the family. That's the reason you need to report. So they have a guideline for that. That's what our definition for the germline mutation. And also, we also, from this study, we want to identify what is the somatic mutation. That means it's specific for the tumor. It's not in the somatic cells. So our definition is genomic alterations that change the patient's diagnosis or may target by the FDA drugs or in context of existing clinical trials according to the NCI adult match trial criteria. So there's an ongoing NCI med trial is doing the same thing. They're using these genomic tools to identify specific mutations in tumors and then put it into the right category for this trial. I think the previous speaker also allude, now they even in all sorts of cancer, right? The previous speaker is talking about the lung cancer. You want to know what the specific molecular profile of this cancer so that you can treat it with the right drug or methods. So that's the idea behind it. So this is a data overview. This slide is a pretty busy slide. It's not meant for you to see the detail, but I gave you an overview of this thing. And this category is identified by RNA-seq. Basically, these are all the fusion genes we detected in those tumors. You can see there's a lot of fusion. It's diagnostic for the tumor, okay? Especially with the pediatric cancer. For example, the Ewing sarcoma, you have EWSR, FLE1, or ERK fusion. This is all the Ewing sarcoma. And you have this SIK FOXO4, which is the Ewing-like sarcoma. So all this, I'm not going to go each one, but this is a very diagnostic marker for the tumor. And also their drivers. And this part is the DNA sequencing. From the DNA sequencing and RNA sequencing, basically this is somatic mutation we're detecting in all the tumors. And those are the driver tumors, and some are actionable. And here is the copy number change detected by the SNP array. And some copy number change genes, it's also is actionable. And this is also from the DNA germline sequencing, we can detect the germ disease causing or actionable, and also have the consequences with the family members. And this slide shows that using this technology, this kind of platform, we can accurately diagnose the cancer. With these 4 cancers, because of the genomic study, we changed their diagnosis. For example, this EWS original was a Ewing sarcoma. They think it's a typical Ewing sarcoma, but we detect a fusion gene that is not the typical Ewing. So it's detected. It's changed the diagnosis as a Ewing-like sarcoma, and the same with the other cases that either the expression profile is not as original tumor, or the hallmark fusion is not there. So we have to change the diagnosis. And in terms of the germline mutation, we detect about 10% of the patients in this cohort has actionable germline mutation. You can see some germline mutation is pretty serious. You have BRCA1 mutation, you have TP53, which is a Li-Fraumeni syndrome, that would predispose for other cancers, not only for the patient, but for the family as well. So this is important information for patient to know, and for the treating physician to know too. And on the somatic side, we detect about 50% of the case have actionable somatic mutation. And you can see this is the mutation we detected. And here is the drug. Potentially, we can use those drugs, FDA-approved drug, to treat those mutations. So this is a very important thing, is all this discovery is readily to be used for those patients. So in summary of this study is we demonstrate use this study the importance and the feasibility of performing multi-dimensional clinical clinomics in the clinical setting in the real time. And 50% of children having this actionable somatic mutation, and 10% has germline, actionable germline mutation. So this is, after this feasibility study, this is the model we think how to use genomics in the precision medicine. You know, in the metastatic disease, all comers, they might have different molecular profile. By using the genomics to identify the biomarkers, we can separate them into some people have good signatures, some patient has poor signatures. But a good signature, we can use standard therapy, which we know they're going to respond to the standard therapy. So we just give them the standard therapy. But for the people who had a poor signature, we can further detect their true driver mutation, and then can use targeted therapies to treat those patients. So hopefully that's the way we can probably improve the survivability of those patients. And the CCR currently established a clinical program based on our study and this kind of hypothesis. And when the patient come into the CCR, the Center of Cancer Research, usually people come into, the cancer patient that come into the CCR, they are not a regular cancer patient. They are all relapsed. They exhausted all the possibility standard therapy outside, right? There's nothing they can do about it. That's the reason they come here for clinical trial. So these are all high-risk patients that come to the CCR. And those patients that we're going to, different PI, depends what kind of cancer they enroll them into different clinical program protocol, and they can order this next generation sequencing test. And the biopsy was sent to the pathology. They do the nucleic acid extraction, and they do the initial QC, and they send it to us to sequence. This is all clear certified process. And from that DNA, RNA material, we can get the germline data and the somatic data, and can have the tumor board to discuss what is the important mutation found in this tumor. And the genetic board, we can identify the germline mutations identified in this study. And eventually those records will put into the clinical medical record. And also the data is available for the clinical research, which is stored in the BTRACE database in NIH. So the whole process takes about, from our pilot study, is about a 4 weeks turnaround. That's a pretty fast in terms of all these kind of things. So we can use this program to really benefit the patient. And this is the program, the instrument we use in the program. We have two NextSeq 500, and the reason we use those is the high speed. They can sequence about 65 gigabase in 14 hours. It's very fast. Overnight, you can sequence all this thing, couple of patients using this instrument. And at the same time, we have a HiSeq 2500, which has a much higher throughput per run, about 1,000 gigabase per run. But it takes longer. It takes 2 weeks to run one run. And this pie chart shows up to, I think it's the end of August, we have sequenced 255 patients of 81 different diagnoses of the different cancers. And another important thing, the tool we developed is this data portal. You can imagine this technology generates a lot of data. And it's not easy to look at those because those are just like a short reads, right? It's bioinformatically, there's like terabytes of data. But if you are a biologist or a clinician, it's no way for us to see the result easily, okay? That's the reason we built this tool to visualize the data from the website. And this is the front end of the website. In here, this is the patient summary page. You can see this patient will have a case, which is a biopsy, bear this code. And we have like a five experiment for this biopsy. We did an exome sequencing. We did a panel sequencing of the N stands for the normal, which is the blood sample. And the T is for the tumor. This is the tumor sequencing. And the T1R that represent the RNA sequencing. So we have exome panel sequencing for both germline and the tumor DNA, as well as the RNA sequencing. And this is the coverage plot, how good the sequencing is, and how this variance summary. And we also developed this QC report. We want to see how good the experiment that we did, right? This one gave you an overview of the QC, how many reads we generated per library, and what's the coverage for this sample. And another important thing is we perform genome typing every time for every patient. Ensure there's no messed up in the lab to swap samples. So this is all genome typing using the base pair resolution sequencing data. And this is further QC. We can see this is a circus plot. We plotted the variant allele frequency. You can see this tumor, lot of loss of heterozygosity. So it become homozygous in a lot of regions. This is all chromosome, 1, 2, 3, 4, all the way to chromosome 22, and the X chromosome, Y chromosome. And this just give you an overview of the genome. So this tumor apparently has a lot of aneuploidy, has a loss of chromosome. And we can estimate what's the tumor content, how good your biopsy is. In this case, the tumor is about 45–50% tumor. The rest is normal cells, so we can detect that. And in the RNA sequencing, we can detect how good the RNA quality is. This is from 5' end to 3' end, what's the coverage, right? And here is the hotspot coverage. So those are most important disease-causing variant that we need to know how good our detection is. And this page just give you all the germline and the somatic mutation. If you click different tab here, you can see the different information. And here, this slide show you example of somatic mutation we detected in one non-small cell lung cancer patient in the tumor. This is a classic T792M mutation in the EGFR gene. This mutation is confirmed to the EGFR inhibitor resistance. So if we know this patient bear this mutation, and we will predict that the EGFR inhibitor is not going to be effective for this patient. So then immediately the clinician will know that what things they should use to treat, what drug they should not use to treat this patient. This one just show you the copy number of the tumor. This is a 2 copy, the genome diploid genome 2 copy. Anything above it is a gain. Anything below it is loss. You can see this tumor has a lot of copy number changes in the tumor. And another thing we developed is the mutation signatures. This is developed by the Sanger. The context of the mutation, how the somatic mutation happen in the context, because this will give us a hint how those mutations arise because due to the chemistry of the mutation, sometimes it's because of the repairing mechanism, and sometimes it's because the UV or other things. The carcinogen, the signature is going to be different. In this case, in this tumor, we detect that there's an enrichment of the signature 7. If you look it up in the Sanger Cosmic website, the signature 7 is a UV signature. So this is a melanoma and has a typical UV signature. And we also have a tool for the mutation burden. We calculate how many somatic mutations per megabase. This is very important for nowadays immune therapy, because all this immune therapy, there's one biomarker is this mutation burden. If you have a higher mutation burden, usually they are more likely to respond to the immune checkpoint inhibitor therapy. But otherwise, if you have a low mutation burden, likely you're not going to respond to that. And we also developed a tool to visualize the fusion genes. You can see this one is the EWS WT1 fusion. And this is the fusion partner EWSR, the front part, fused with WT1, the later part, and they created this fusion gene. And this gives you exactly the protein sequence as well as the nucleic acid sequence. That's very easy using this tool to design primer to validate this fusion. So there's other tools we developed on this website, including the tissue typing, the HLA typing, new antigen prediction, gene expression, and the GSEA analysis. And also, if we have the survival data associated with the samples, we can perform survival analysis. So in conclusion, the next generation sequencing, including the whole genome, exome, transcriptome, and determines the complete genomic and epigenomic portrait of the cancer at a base level, base pair level. So it's a very high resolution information. Integrated analysis of cancer can identify the biological relevant diagnosis, prognosis, biomarker, and also the novel targets for the precision medicine. So this is a huge teamwork, and the Clinomic project, basically it's the collaboration between the genetic branch and the laboratory pathology. And there is a lot of individual contributing into this program. There's a lot of wet lab biologists and the bioinformatics, because this genomic is highly reliant on the bioinformatic tools. We develop new tools, I show you those, and also the people's dedication to make this happening. And also I want to acknowledge the CCR leadership to make this program possible. Okay. Okay.
Question?
Yes.
When in the cancer care process would this become possible? Like, so is it only for somebody who has metastatic tumor, or would you recommend it for somebody early on? Or how would you decide when to implement all these genomes tested for patients?
It really depends. In the CCR, as I said, in the CCR, they all coming, all the patients are high risk and the metastatic. They don't have any way to treat them. In the past, before the genomic era, people just guess. You try this, doesn't work, you try the other one. You just shoot in the blank, okay? Now we have these two, they have some rational, educated guess, okay? If they have the mutation, they maybe match up with the inhibitor or something. So that's the utility. But the question is, now this kind of study is still pretty expensive. You know, we spend about like I think anywhere between 3,000 to 5,000 per patient for this kind of thing. So I think this sequencing, the price eventually coming down, they will be very, if it's cheap, it's okay. You can just order it to do it. But currently within the CCR, the idea is still using this for, because those patients are all high risk, the idea is we just sequence everyone. But outside the NIH, I'm not sure. You know, I know some pediatric cancer, the insurance even covered this kind of study. You know, but I'm not sure if it's localized disease or those kind of things. I'm not sure if the insurance how common they use this too. Yeah, but eventually they will use it because it's all about the cost now. Yeah.
So the precision medicine is pretty well defined for adults. But for the children, if you identify a mutation, you can't necessarily effectively use the adult drug.
Well, that is true. I think now the bottleneck is the drug availability to those mutations. A lot of mutations we found, 50%, we just don't see. We know probably it's a driver, but we don't have any effective drug. Another thing is this kind of adult drug. How do you use in the pediatric case? I'm not a clinician, but I think they are working out these things. They have clinical trial to try those drugs or repurpose some drugs for this kind of use. Yeah.
Okay, that'll do it. Thank you,
Okay,
John.
thank you very much.